Welcome back to part 5 of our video on RNA and DNA. Now we are focused on translation. We have looked at how RNA is made from DNA in a process called transcription in part 4. Now we're looking at once we have made that RNA, once we have made that message, how do we turn that into a product? Once we have the message of what we want to build, how do we read that in order to turn into a product? How do we translate the message, translate all these series of letters into a product, into what we actually want? So we have made the RNA. The mRNA has left the nucleus and gone to an organelle called a ribosome. This ribosome will help make the product, which is called a protein. First off, once it reaches there, it has to know how it's going to be read. And we read it in 3-letter words. And we call those 3-letter words codons. So you separate the entire series of letters into 3-letter words, 3-letter codons. So AUG, GCC, AGG, CUU, AGG, CAA, and UGA in this example. When it reaches there, it must then have, the ribosome must read the code and match up what needs to be built. To do that, we finally get to what we mentioned in part 3 of our series on DNA and RNA. In video 3, we talked about tRNA. Now we're going to look at what tRNA is. It is called tRNA because it is transfer RNA. It transfers the product into the mRNA. It transfers it where it needs to go so it can be built. The product that it is transferring is called an amino acid. And how it knows what amino acid to put in what order is it reads this codon and looks at what matches up to the codon. Well, the tRNA has a series of 3 letters as well, called the anticodon. And the anticodon should be the opposite of the codon. So let's start with our first codon. The anticodon is only this series of letters. Everything else that is attached to it is the tRNA. This first series of letters, we have our codon of AUG. So we're going to come in with our tRNA, and we're going to match up the anticodon that goes with this. So your ribosome will look through its tRNA, and eventually it will find, well, we have AUG. Well, I happen to have this tRNA that has an anticodon of UAC. Well, the U matches up with the A matches up with the U, and the C matches up with the G. So that must be what the first amino acid that this sequence wants is. And so it matches up, and it happens that our first amino acid is an amino acid called methionine. It then goes to the next one in the series, the next codon, GCC, and from here on, I'm just going to be writing the anticodons so that I don't have to waste time drawing all this. So what the anticodon for this would be CGG. And that one is an amino acid called alanine. And so the methionine and the alanine will now join together and start making a chain of these amino acids. And so you'll have a long chain of all these amino acids put together. All right. The AGG will match up with UCC. And when we look at that amino acid, we find it is an amino acid called arginine, abbreviated ARG. And so it gets added and attaches. Then we go to CUU. We match up that CUU with GAA. And when we match it up, one thing that I forgot to mention initially, when you are matching these up, because when you have to do it, you will use that green circle chart. When you're matching them up, match up the codon. Do not match up the anticodon. When you're trying to figure out for this codon, CUU, well, what amino acid do I put here? Do not look up GAA on your chart. Look up the original codon. CUU is what decides what is up here. So CUU, if you run your chart, will match up to an amino acid called leucine, LEU. And then we go on. AGG will match with UCC. And when you read the AGG, you'll find out that it is, again, a second arginine, ARG. CAA will match with GUU for the anticodon. And the GUU anticodon tRNA will have what CAA is asking for, and that will be glutamine, GLU. And then we reach UGA. Now, UGA is one of three special codons. Because when you read it on your chart, you'll notice UGA codes for a thing called a stop codon. This is a stop codon. That tells the ribosome, we are done building what we want to build. It is functional. This is what we want to build. Thank you for your time. Please release the protein. So, it releases the protein, and the protein then goes flying off, and final things are done to it, which we don't need to talk about, to prepare the protein for doing its job. But once you reach this stop codon, your protein is done. And we are done with translation, the process of making that protein. This entire thing, or this entire thing, is the protein. And proteins are made up of chains of amino acids. So each of these green things is a different amino acid. The entire thing together, the entire chain of amino acids makes a protein. And that is what the mRNA is trying to transmit with its code. So that should be it with translation. We have made the RNA. We have translated the RNA into a protein. We are now done with DNA and RNA. in making the DNA, what it's built of, RNA, what it's built of, and transcribing the RNA, transcription, where we take DNA and build it into RNA, and translation, where we take that RNA that we have built and turn it into this protein. Part 6, the final part of our DNA and RNA, will be on what happens when making the DNA during either the DNA replication or during the transcription where we make the RNA, or in another instance, what happens if the DNA itself is just damaged. And the code, something changes about it. The DNA is damaged and it is broken, and something changes in it. That is called a mutation, and that will be the subject of video number 6, part 6, where we talk about DNA and RNA mutations and what effect they will have on what we have done here.